Okay, good afternoon. My name is Benjamin Deonovic. Today I'll be presenting on haplotyping and quantifying allele-specific expression using RNA sequencing data. The layout of my presentation is as follows. I'll give a little background. I'll describe the problem that we're trying to solve, and I will describe the two models that we have set up to solve this problem. So diploid organisms contain two copies of their genetic material, one paternal in origin and one maternal in origin. There exists genetic variation between these two strands. And I have a schematic here. So here we have a simple gene where I've shown these two chromosomes, one paternal in origin, one maternal, and I've identified four loci on this gene where there's variation between these two. So here you can see this one has TCGC and this one CGTA. Now this combination of variants is known as the haplotype. And these variations result in alternative forms of genes referred to as alleles. Now it turns out that these alleles can be regulated and expressed unequally, which is termed allele-specific expression. And so again, in this schematic, if in the cell there can be a certain proportion of genes that are expressed from this strand, we'll call that proportion rho, for example, and then 1 minus rho proportion would be expressed from this strand. Now the problem that we're trying to solve is to phase the haplotype. So that is to figure out what the combination of variants is. So using data, it's very easy for us to figure out where the variants are and what the possibilities are. So it's very easy to figure out at this loci in this gene, there's a variant, and some people in the population have a T and some have a C. It's much harder to phase the haplotype, to figure out that T is followed by a C, and then a G, and then another C. Secondly, what we want to do is to quantify the extent of allele-specific expression. That is to estimate that rho proportion. Furthermore, because genes can have multiple isoforms, that is due to alternative splicing, this gene might express an isoform where this middle exon is skipped, for example. We want to see if there might be heterogeneous isoform ASE. That is, isoforms in a gene might have different proportions of rho for each isoform. To this end, we have developed two models to try to solve these questions in our software named IDP-ASE. A little background on some previous software. I have listed here some software that is aimed to analyze this ASE problem. But there's two major drawbacks to these methods. Many of them require family trio data. That is, they require sequencing data not only for the individual you're interested in, but also their parents. So that is obviously laborious and more costly to obtain. Or some other methods might require the haplotype to simply be known and to be given as input. And based is one of the only methods which is capable of obtaining gene-level estimates of ASE using RNA sequencing data alone. So we'll be taking a special note of this software. Now next I want to talk a little bit about the data. So a lot of the previous methods I have on the previous page use what's called second-generation sequencing data, SGS data, or sometimes referred to as short-read sequencing data. The two major aspects of this data is the low error rate, which is a good thing. However, they have very short read lengths, about 100 to 150 base pair. And this means that a single read is often incapable of covering multiple variants, which is a problem if you're trying to phase the haplotype. If you don't cover multiple variants, it can be very difficult to determine what the haplotype is. Now, there are methods that utilize what's known as third generation sequencing data, or TGS data. Sometimes we'll refer to this as long reads, which are of much longer read length, about 1 to 100 kilobase pairs, and these are able to cover multiple SNPs. However, TGS data has much higher error rate than short read sequencing data, which can limit the accuracy of haplotyping, and also has much lower throughput, and as it turns out, is not suitable for quantifying ASE, as we'll see in a few later slides. Now, one of the innovations of IDP-ASE, other than the model that we use, is utilizing hybrid sequencing data. That is, combining short read sequencing data and long read sequencing data to obtain the best of both worlds. And so just to do a quick little summary here, IDP-ASE is the first method to simultaneously phase haplotypes and quantify the extent of ASE at both the gene and gene isoform level. And we do this using these two models, which I'll briefly describe here. For the gene level, we use a Bernoulli mixture of categorical distributions to phase haplotypes and estimate ASE at the gene level. And we fit this model using MCMC. And for the gene isoform level, we will extend the Poisson model for RNA sequencing isoform expression estimation to estimate the heterogeneous isoform ASE. So to begin, I'll describe the gene level model. So I have a lot of notation here, but I think the picture is really where you can get a lot of information. So to begin, suppose we're considering a gene loci with M single nucleotide variants with known locations and variant calls. So these variants can come from software such as SAM tools, GATK, using RNA sequencing data only. Now suppose each variant site is biallelic and heterozygous. So at each variant, it can either be an A or T, a T or G, etc. And they're never homogeneous, they're never just A or A. So here, I have the same gene schematic I had before. Here we have our two genes. And this right here is what you would get out of using something like SAM tools or GATK. You would get the variants and the variant calls. So here I'm referring to W0. SAM tools might call that the reference, W1 as the alternative. So here we have C or T, C or G, G or T, A or C. And now in our model, we refer to the haplotype. So this haplotype up here, the H, is something we do not know. This is something we're trying to estimate. It's unknown to us. And you can see here, the haplotype for this blue strand is 1001, corresponding to TCGC. And similarly for the other strand, 0110. So that's what's described in those words there. Next, I describe this, what we call the read matrix right here. So suppose we have RNA sequencing data, which is represented here by these lines. We have short read data and long read data. For the read matrix, we look at the variants that each read covers, and then we fill in the matrix based off whether that corresponds to the reference or the alternative. So here, for example, if you look at this long read right here, corresponding to the third line, or let's use this fourth long read right here, corresponding to the fourth line, you can see the variant calls on it are TC, GC, so we get 1001. Now this immediately shows you the great benefits of using long read sequencing data. That single long read gave us the exact haplotype that we were looking for. However, a problem with long reads is often they might have higher error rates. So if we look at this third long read right here, it actually has a little error here, something we wouldn't be expecting. And we've denoted this by a 2 here. It doesn't match either of the variant calls from SAM tools. Now let's take a look at the short read data in the read matrix. You can see the short read data is not very helpful for figuring out the haplotype. The short read data very often only is able to cover one variant. And so you're not able to get the linkage between the variants to figure out the haplotype. So this read matrix is one of the inputs to our method. Each row of the read matrix we fit a model to. We fit it as a categorical distribution with a parameter p right here, where Q is a user input or data-derived error model. Now the model is general. As I said, it accepts reads from various kinds of sources or platforms, be it SGS data, TGS data. And the error model is also very flexible. In our method, we utilize the quality scores from the sequencing data. And the goal of IDP-ASE is to estimate the 2 parameters in our model, H and rho. And so we want to fit a model to our data, F, some kind of likelihood function, and try to maximize that likelihood function to obtain estimates for our parameters. So we adopt a Bayesian framework here. So using Bayes' rule, we get the model is proportional to the likelihood of the data times the prior of the parameters. We'll use a non-informative prior for the joint distribution of H and rho to simply get the likelihood over the data. And now if we suppose the reads here, are conditionally independent given the haplotype and the error model, we simply get a product over the data likelihood. And to account for ASE, we model each read as a mixture distribution where the mixture weights are rho and 1 minus rho, corresponding to the haplotype. So here this F is simply the categorical distribution given right here at the bottom. And it's a mixture of using the haplotype H and using the corresponding haplotype H bar, which is simply 1 minus H. As I said, we use MCMC to obtain samples from the posterior distribution, which we then use to obtain maximum a posteriori estimates of rho and H. We use slice sampling for rho. We use a Metropolis Hastings type of sampler for the haplotype. The MCMC samplers initially run for 1,500 iterations. 1,000 of that is used as burn-in, and convergence is determined by the Gelman-Rubin diagnostic. If the model looks like it on the rho parameter. If the model looks like it hasn't converged yet, we continue to do 500 iterations at a time until convergence has been met. And now that after running the MCMC, we have estimates of what H and rho are. And so we want to evaluate how well our model is able to estimate these 2 parameters, H and rho. To evaluate the performance of estimating the haplotype, we look at GM12878 data, which is kind of a gold standard, which has been extensively sequenced, and the haplotypes are known. We use 2 metrics to look at the performance of our model, CHPR and CSPR. CHPR is the correct haplotype phasing rate. So this means what proportion of the haplotypes we have exactly phased correctly. So if a haplotype is 0, 1, 0, our model outputs 0, 1, 0, it got it correct. The haplotype 0, 1, 0 and our model outputs 0, 1, 1, we did not get it correct. The second metric, CSPR, the correct single nucleotide variant phasing rate, looks at what proportion of the variants did we correctly phase. So if the haplotype is 0, 1, 0, and we output 0, 1, 1, then we've correctly phased 2 out of 3 of the variants. To evaluate the performance of IDP-ASE at quantifying allele-specific expression is a little harder because there's no gold standard data to do this. And so we rely on simulation data instead. And so we utilize the structure of the read matrix obtained from the GM1287 data to get simulation data that has coverage and number of reads similar to real data. But we simulate the haplotype and the row parameter and then fill in the read matrix accordingly. The haplotype is simulated simply by flipping a coin, zeros and ones. And the row is simulated from a truncated normal distribution with parameters set to match what we've seen from real data. So this first plot up here on the top is data from the GM12878. It is the distribution of the number of variants within genes. So this plot shows the importance of utilizing a method like IDP-ASE. As you can see, a lot of genes only have one variant in them, about 40%. However, that means 60% of genes have more than one variant. This indicates that if you use short read data only, it's very difficult to phase the haplotype correctly because those short reads are only able to cover one variant. And so this plot shows the importance of using either third-generation sequencing or hybrid sequencing. Next, this plot shows the performance of our method on phasing the haplotype on GM12878. In this plot, we have the CHPR on the vertical axis. This plot, we have the CSPR on the vertical axis, with number of variants in a loci on the x-axis. The hybrid sequencing data is in red. Short read sequencing data is in the blue. IDP-ASE is in the solid lines, and M-based is in the dashed lines. First thing we can see is clearly IDP-ASE using hybrid sequencing outperforms all the other modes, able to correctly phase the haplotypes at a very large percentage. Next thing that we can note is that there's very little difference between M-based using hybrid sequencing and second-generation sequencing. This is because M-based only utilizes the marginal counts of the read matrix. It doesn't utilize the linkage within the read matrix, and therefore it's not able to utilize the information that the long reads give you. Next, we see that IDP-ASE is able to utilize that information from long reads, as we can see from the performance improvement jump from using second-generation sequencing to hybrid sequencing for IDP-ASE. And finally, you'll notice using IDP-ASE, you can also use it with just short-read sequencing data, and it will perform similarly to other methods, such as M-based. The story is very similar for the correct single nucleotide variant phasing rate, as you can see from this plot. And you can see that IDP-ASE is able to correctly phase about 90% of the variants, no matter how many variants we have in a gene. So next, we also want to investigate the effects of coverage on the haplotype performance. And so coverage is the number of reads, short read or long read, that map to a gene. So to do this, we plot the CHPR and CSPR against the read coverage. Here we use adjusted read coverage, and I'll explain what those are. For the long reads, the adjusted read coverage is given here, where N is the number of long reads that are aligned to loci. so that's simply the coverage. N is the number of variants in the loci, so we divide by the number of variants in the loci, because we expect genes that have more variants to be longer, and therefore to have more long reads. So that's kind of a normalizing factor. And C here is the maximum number of variants that a long read can cover in this loci. So note here, simply C over M is simply a multiplicative factor between 0 and 1. Short read, adjusted short read coverage is similar. It's simply N over N. So in the next two plots, genes have been grouped by the 10 percentiles of the log-adjusted coverage, and a smooth low S-curve is fit to the data. So here on the top, we have long read coverage in the first row, and short read coverage on the bottom row. And the CHPR in the first column, CSPR in the second column. What we see here is that as we have more long reads, the performance of IDP-ASE improves, whereas the performance of M-based does not improve as the number of long reads increases, which similar to the point we made earlier, that shows that M-base is unable to utilize the information the long reads provide. In this bottom plot, we see a similar story. As the number of short read increases, the performance of IDP-ASE increases as well. This might be simply because the number of short reads is correlated to the number of long reads. And so as short reads increase, the number of long reads increase as well. But again, we see that N-based and also short read sequencing only data does not have very much improvement as the coverage of short reads increases. These plots are to show the performance of IDP-ASE for estimating the row parameter, the extent of ASE. So here in this plot, we have the Euclidean distance of the estimate to the true row plotted against the number of variants in the gene. You can see here IDP-ASE, the solid lines, tend to dominate the corresponding dashed lines of M-based, indicating IDP-ASE has lower errors, which is better. Next, we see that hybrid sequencing, again, gives us the best performance for estimating row versus short read only or even long read only. Long read only here has the worst performance of them all, indicating, as I've said earlier, that long reads are not appropriate for estimating ASE on their own. On the right here, we have the density plots of the estimates of the row. The gray dots here indicate the simulated row density. It's a normal distribution. And you can see the output of IDP-ASE matches very closely to the simulation, whereas M-based is very shy about estimating the row to be 0.5 and tends to favor values just close to 0.5. Now the model I've described above is for the gene level estimation of the haplotype in the ASE. But as I mentioned, there might be, the data may contain a mixture of gene isoforms that can have heterogeneous allele-specific expression. So let's suppose that the gene that we're interested in has k different isoforms. Assume each isoform contains a variant, and let theta k be the expression abundance of each isoform. So you might obtain estimates of theta k using something like cuff-links or RSEM or our IDP software. Now this theta k can be further decomposed into theta k0 and theta k1, which are the allele-specific abundance of isoform k. So again, a picture does a lot better at describing this model. So here we have, again, the same gene I described earlier, but we've also indicated a possible isoform of this gene, which has exon skipping of the middle exon here. Also, you'll note that this exon 1 has a variable start site. So let the regions, define exon regions as a non-overlapping set of exons that comprise the isoforms of the gene. So you can see the regions indicated here, R1, R2, 3, and 4. Suppose there are S number of regions and junction regions that can span multiple of these regions. So junction could be between R2 and 3, or perhaps between R2, R3, and R4, indicating that a read could potentially map to this region, and that region. So there's S, total number of regions or junctions like that. And define LS as the effective read length, as the effective region length, which is defined as the number of positions from which a read can map to that region. We'll have n total number of short reads that map to this gene. Now, if we assume that read sequencing is a simple random process in which every read is sampled independently and uniformly from every possible position, we can derive the Poisson model for isoform abundance estimation. So the read count at region S, we'll call it YS, can be modeled as a Poisson random variable with parameter lambda s, which takes this form, where m is the total number of reads in the gene, ls is the effective read length, alpha skj is 1 if region S is contained in isoform K under the corresponding haplotype, 0 otherwise, and theta kj, as I've mentioned earlier, is the abundance of the K isoform under the corresponding haplotype. And so, since the Poisson likelihood is concave, we can simply use something like the Newton-Raphson algorithm to obtain the MLE for the theta kj. Now, once we have estimates of the theta kj, let's say it's theta hat k0, theta hat k1, we can use these estimates to get an estimate of the isoform-specific allele ASE. We'll call that tau k. That can simply be estimated as this. So that's what our gene isoform level model does, and it gives us estimates of the gene isoform level ASE. Next, we'll look at a simulation to evaluate the performance of our gene isoform level model, and then finally we'll look at a few examples that showcase our model. So here's a simulation where we have simulated the theta k values to see how well our model performs when there's no allele-specific expression. So here we have the true value of the FPKM, that's the theta k, and the estimated FPKM. You can see there's very high correlation, indicating that our model is able to recover the simulated value. This plot shows similarly the true tau versus the tau hat that our model would give you. And you can see, although there's very high correlation, the data is a lot noisier, indicating the difficulty that there is in estimating gene isoform level ASE. But as you can see, our model is still able to give you a very unbiased result that's highly correlated to the truth. So next, a few examples. Our data is run on real data from MCF7 cell line breast cancer and a human embryonic stem cell line, the H1 cell line. This gene exhibits the utility of using our method for phasing the haplotype. Here we have the number of reads that have the corresponding variant calls. And what a lot of methods do is called pseudo-phasing, where they simply look at the reads that had the variants that had the most reads, they get phased together. So something like M-based might phase the C and the G together. However, in this read, we have 75 long reads that support the haplotype phase that our method outputs, which is TG and CA. Here are density plots from, in this first column, the MCF7 cell line data, and in the second column, the human embryonic stem cell data. On the top, we have the gene level rho estimate. On the bottom, we have the isoform level tau estimate. You can see here the difference between the breast cancer cell line and the human embryonic stem cell line. There is much larger variance in the breast cancer cell line with a lot more what we would call ASE genes. Here in the red, we have indicated genes or isoforms that have rho or tau below 0.35 and above 0.65. And finally, we have a few more examples that indicate the complexity of allele-specific expression and isoform expression that can exist within an organism. So in this first gene, we have an example of, let's start with this second gene here. We have an example of SNPs that are not common to each isoform. So here we have a variant that only is exhibited in this isoform, and here we have a variant that only exhibits this, that's only exhibited in this isoform. On the top here, we have a variant that's shared between these two isoforms, another variant that's shared between these two isoforms, but we have reads that are able to distinguish because they map to junction regions and we're able to distinguish these two isoforms. Here we have a variant that's shared, one that's only on this isoform and one that's on the other one. And finally, again, these variants here can be distinguished and correspondingly their isoform level ASE can be corroborated by these junction regions, reads that map to junction regions. And so again, to reiterate, we have developed a method, IDP-ASE, which is a novel method that is able to phase haplotypes, that is able to quantify gene level and gene isoform level ASE using only RNA sequencing data. Thank you.